Hello, everyone, and thank you for attending today's webinar, Redefining Antibody Specificity Profiling for IND. I'm Rebecca Cushmiter. I'll be hosting today's webinar. I'd like to start by introducing our speakers. We will be joined by Chris Raghavan, PhD. He's the application scientist at Integral Molecular, and Diana Norden, PhD, the senior scientific writer at Integral Molecular. You can read their full bios on the left side of your window by selecting the Speakers tab. Just a few notes before we begin. To access additional resources from today's presentation, you can click the Handouts tab button on the left side of your screen. You can access closed captions from the bottom right corner of the video player. This webinar is being recorded. It will be available to watch on demand within 24 hours. There will be 3 audience polls throughout the presentation today. We hope you will participate in all of them. And we would love to hear from you, of course. During the presentation, you can submit any questions you have using the Q&A tab on the left side of your screen. All right, let's go ahead and begin.
Thanks, Rebecca. Hello, everyone. I'm really happy to be here. Today, I, Chris Raghavan, and my colleague, Diana, are here to representing Integral Molecular all the way from Philadelphia. Just to give you a little bit of intro about our company, a lot of people often hear small biotech and think startup, but no, in fact, we've been established in the industry for over 20 years. We started as a spin-out from the University of Pennsylvania, and since then, our expertise has centered around membrane proteins and antibodies, as I will for sure get into. Our talk will be broken into 2 main sections. The first, introducing you to the Membrane Proteome Array, or MPA, as you will be hearing that acronym very frequently, our role in drug development, and a breakdown of our project pipeline. Later, Diana will touch on the MPA's relationship with regulatory bodies like the FDA, and some case studies from customers that have successfully used the MPA in IND submissions. We will, of course, have some Q&A at the end, but for now, I will get into the presentation proper. As I'm sure you're all aware, the road from antibody discovery to clinical development is long, expensive, and involves a great deal of risk. So we provide the MPA as a service to help de-risk the early development of your molecules. We have tested over 1,000 molecules on the MPA, and we have found that about 1 in 3 of these molecules are polyspecific. And of course, specificity screening is essential for identifying potential off-target protein interactions, which can be responsible for dangerous side effects, as we have observed from commercially available small molecules that have had major side effects or have been withdrawn from clinical trials. Of course, discovering that your lead molecule produces off-target effects so late in development can be a program killer. Thus, the MPA exists to prevent those types of outcomes and de-risk drug discovery campaigns early in preclinical development. So to address this global need for better preclinical tools, we developed the field's leading solution for specificity profiling. We are proud to have been trusted by over 100 companies that have incorporated the MPA into their drug development programs, further validating our data in their studies published in high-impact journals taken into the clinic. You will hear more about these types of studies later on in this talk from Diana, but first I want to get into, really dive into the technology that makes the MPA unique. The Membrane Proteome Array is a platform to screen roughly 6,000 natively expressed membrane proteins. We do this in a 384 well-plate format using transiently transfected HEC293 cells. What we feel really separates us from our competitors is that we measure target binding on a highly sensitive flow cytometry platform, and more importantly, we are the only platform that screens against native epitopes in unfixed cells, so as that we can be more representative to how these protein targets will be expressed in vivo. Now, using these methods, we're able to profile the specificity of your molecule for its intended target and identify any off-target binding that may exist. To get into what is included in our library, our protein array has about 94% of the human membrane proteome coverage. This includes transmembrane proteins, ion channels, GPCRs, transporters, and more. We have also added a heterocomplex library that has been highly curated to include proteins that may need to be expressed as a complex to facilitate binding that would be expected in vivo. So things like integrin complexes or TCR complexes, we have that as well. And please reach out because we would be happy to share the full list of proteins included in our library. Now, our platform has been successful with a wide variety of molecules and modalities. We specialize in monoclonal antibodies, but we have also had a lot of experience screening scFvs, bispecific antibodies, nanobodies, and even peptides. Our platform is also appropriate for ADCs and CAR T applications. And customers with successful IND experiences have also shown that for CAR T applications specifically, testing just the scFv portion can produce the most time and cost-efficient specificity profiling while still being in accordance with FDA guidelines. Now, I'm going to move to highlight some of the advantages you will get when you're using our platform. The first being a diversity in cell lines to improve the resolution of our screens. This is important because some protein targets that are endogenously expressed in HEC293 cells, in this case B7H3, can result in a high degree of background noise that makes the screening of this molecule unfeasible. As you can see here, there is just so much noise. By using QT6 cells, which come from quails and are evolutionarily divergent, we are able to significantly reduce the background signal and improve the sensitivity by which we can detect target binding. During the initial optimization stages of our projects, we will test your compounds in both cell lines to determine which is the optimal condition to proceed to screening with on the full array. Now, another key benefit to our platform that I touched on earlier is that we are the only technology conducting full library screens in unfixed conditions to really maintain the physiologically relevant binding conditions that you would expect in vivo. I am sure you are all aware that fixation can alter the native conformation of certain epitopes. This type of fixation induced changes to membrane proteins can cause false negatives by preventing antibody binding, or even false positives from artifactual binding. Understandably, this can be a huge issue when testing molecules for binding specificity. To further emphasize this, I will showcase this in-house testing that we did, where we conducted parallel screens comparing unfixed and fixed binding conditions. We tested the same antibody that targets CD19 using our standard of unfixed conditions and found that it had a significant off-target binding interaction. However, when we tested the same antibody in fixed conditions, we did not identify this off-target, really proving to us that the screening for protein binding on fixed cells can really miss these critical interactions. And this further supports the FDA's guidance that against, the FDA's guidance against specificity profiling done in fixed tissues. So that being, the finer points of our technology, but what does an actual project look like? I'm going to walk you through very quickly what the pipeline of our projects look like and explain how we go from receiving your molecule to performing the specificity profiling, sending you results, and more. To begin a project, we ask for three things from you: a purchase order, a project setup form with information about your molecules, and 500 micrograms of your sample. Once we have these things in our hands, we will begin the project's lead time, in which we will plan out the study and consider any special requirements or risks that may be relevant. After this lead time, we will move to our initial assay setup experiments that can take place, being our assay setup. This stage is set up to determine the optimal conditions to test the sample on the MPA screen. We will test the sample against the positive and negative controls using a titration of 4 concentrations, and we will do this in both HEC293 cells and QT6 cells, as I mentioned. Once we find an optimal cell line and concentration, we will advance to the MPA screen stage. At this stage, we will screen your sample against the full 6,000 protein library for potential binding interactions. Any hits identified are then passed on to the validation stage, which is crucial to rule out any false positive hits that may have arisen. For hit validation, we again titrate the samples against our positive and negative controls, in addition to the hits identified from the MPA screen, to determine what is a true binding interaction and what is not. Now, at this stage, we can also include an isotype control to really rule out any non-specific binding that may have resulted from the antibody backbone or isotype itself. After this validation stage, we will compile the data to be presented in our final report deliverable. Now, at the end of a project, say you find an off-target binding interaction, what should you do next? Well, we often get this question, and we have several post-project services designed to follow up with these results. And our newest one is what we're calling risk analysis, and it aims to provide a more thorough understanding of any off-target binding interactions that may have been identified in the original study. This package includes a more rigorous set of experiments designed to better quantify the EC50 for binding strength, the Bmax for protein expression, and even visualize the subcellular localization of your test molecule's interaction with the off-target protein. We will then put together our highly detailed risk analysis report aimed to provide a comprehensive picture of your molecule's relationship to the off-target protein. In addition to the risk analysis, we also offer other services like plasmid transfer for clones of off-targets identified in the study. If you wanted to perform follow-up studies in follow-up assays in your own labs with consistency in data and reagents. In addition to that, Integral Molecular also has some more in-depth services such as epitope mapping and antibody engineering, which are designed to optimize your therapeutic compound. And it is worth mentioning our referral program with Lonza, who can perform some more functional cell-based assays if you're really trying to determine whether an off-target identified in the study is benign or potentially cytotoxic. Now, we understand that clients are coming to us at really all different stages in their development pipelines. So we offer two main services to accommodate those different needs based on where you're at in your project. As I kind of touched on the basics of, our standard MPA service is best suited for when you have several molecules and you are looking to select a lead candidate from a larger cohort. But if you already have one or two leads and you're looking to screen for off-targets before filing for an IND submission, we, specifically Diana, will author a customized report package ready-made for you to submit to the FDA. Now, this MPA plus IND package also has prioritized timelines, so you don't just get more value in your data and report, but we will also complete the project in half the time. To briefly get into some examples how our customers have used our data, I want to highlight this case study that was done by a lab in the University of Pennsylvania that used us for lead selection in a CAR-T program. They had two antibodies being characterized for potential clinical development, and they used the MPA to screen both of these candidates. And we found that while both molecules hit their intended target, one of them had significant off-target binding. And using these results, they decided to deprioritize the antibody with off-targets, and in the end, the antibody with no off-targets was advanced to the clinic. Another Philadelphia neighbor of ours, Cavaletta Bio, has also been incorporating us into their development processes. With them, we have screened several novel modalities, and they have used our data in successful IND submissions, which we are especially proud of. Importantly, they were granted their IND without tissue cross-reactivity studies, tissue cross-reactivity being the previously established and inherently flawed method of specificity profiling, which Diana will definitely touch more on later in this talk. Additionally, we at Integral have also been able to endorse the power of the MPA firsthand, as it has been critical to the development of a spin-out company, Cell Surface Bio, which specializes in producing high-quality recombinant antibody reagents for laboratory applications. We understand the reality that is far too common for experimental progress to be limited by poor quality reagents. And so to address this problem, Cell Surface Bio uses the MPA to screen and validate our versamabs to produce clean, highly specific antibodies that bind their intended target and nothing else. So again, through the use of the MPA technology, you can trust this antibody and thus trust your data. Just to wrap up my section, there are, of course, many reasons why the MPA should be attractive to you in drug discovery and preclinical development. Our platform is compatible with a wide range of therapeutic modalities. And again, I cannot stress this enough, we are the only company screening against native, unfixed proteins. Critically, our customers have proven successful in their IND submissions without tissue cross-reactivity, and Diana will talk more to that point, as well as dig into our track record with the FDA and regulatory bodies. I will also add that you can work with us directly, but our services are also available through the partnerships listed below. With that, I will hand off to my colleague, Diana Norden, as she gets into her portion of the talk.
Hello, everyone. For the second part of this webinar, I will be discussing using MPA data for improved specificity profiling for regulatory applications. I've divided this talk into 3 sections. First, we will go over some regulatory guidance documents and talk about specificity profiling for IND applications. Then we will do a comparison of the MPA technology to what has traditionally been used, the tissue cross-reactivity study. And then finally, we will look at some case studies comparing MPA and TCR results. So let's get started. FDA has 2 guidance documents that discuss specificity profiling of biotherapeutics. This is the points to consider in the manufacture and testing of monoclonal antibody products for human use, as well as the ICH guidance, the preclinical safety evaluation of biotechnology-derived pharmaceuticals. Both of these guidance documents state that non-target tissue binding may have serious consequences and that cross-reactivity should be assessed. They further go on to say that assessment of cross-reactivity should be performed using human cells or tissues. And this is where the tissue cross-reactivity study got its name and has since been used. I'd like to point out that the Points to Consider document has not been updated since 1997. This is still the current version used today. And I'd also like to point out an interesting sentence. It does say that appropriate newer technologies should be employed as they become available and validated. So already back in 1997, there's this idea and thinking that the TCR assay might not be the best way to truly assess antigen specificity. And recently, there's been some new developments in how the FDA thinks about specificity profiling and the assays that they recommend. So in January of this year, they put out a new guidance document for development of CAR T-cell products. Again, it states that non-clinical evaluation of the antigen recognition domain should include assessment of specificity. And it further goes on to say that unintended targeting of other antigens, which would be an off-target, expressed on healthy or normal tissue is a safety concern that may be evaluated using in vitro or in vivo studies. But this time, it gives numerous examples. It again tells it gives the assay TCR, but then includes other examples of assays like the protein array. So this guidance document now for the first time delineates specific alternative methods beyond the TCR study. Our own protein array, the MPA that we are talking about today, is currently under review to become an FDA-approved drug development tool. We submitted the MPA for inclusion into their iSTAND program. This is one of FDA's drug development tool qualification programs. And we were very excited. The MPA actually became the first technology to be accepted into the program. And since then, we have now been working with CDER and CBER representatives to qualify the MPA as a new drug development tool. We've received feedback on the MPA technology and its uses. We have taken that into consideration in our product development and in the preparation of our qualification plan. We have since submitted our qualification plan for review. It was accepted for review, and we hope to have full approval in 2025. So let's look at some real-world data and how our customers are using the MPA right now. We surveyed our customers and found that about half of them, right around 50%, have used or plan to use the MPA data in their IND filings. We've had numerous successful submissions of MPA data that did not include TCR for IND applications of antibodies and CAR T cells. We've had many successful submissions to the FDA, but also the EMA and NMPA. And about 25% of the customers that we surveyed reported that the FDA directly requested a newer array-based technology like the MPA. So that was our customers, and what is the general toxicology field as a whole thinking about the TCR assay? So this slide shows some results from a survey that the BioSafe Committee Working Group put out. They surveyed 26 different biotechnology and pharmaceutical companies and asked them about their experience in using the TCR assay and how they interpreted those results. Some key results were that 92% of respondents said that the TCR has not impacted the design of their toxicology study. Further, 81% said they have never seen a correlation of TCR data to preclinical or clinical toxicity. They were also asked about whether they had submitted an IND without TCR data, and here 42% said yes, and 31% said that they were considering doing so in the future. And we think, based on our customer experience and the fact that this survey was now completed 6 years ago, that this number now likely is very much higher, and that developers are more and more comfortable with submitting an IND that does not include TCR data. The general conclusion or take-home message from this survey was that the majority of companies believe that the TCR results really are not predictive of in vivo toxicity. So I've discussed TCR a little bit now, and Chris has also discussed the MPA. So let's do a little bit more of a comparison between the two technologies, or the two assays. First, what are tissue cross-reactivity studies for those who maybe aren't as familiar? They are immunohistochemical screening assays used to identify binding of test biologics in different human tissues. Over 30 different human tissues are tested for binding by the molecule, and the staining is evaluated by a pathologist. This assay can give you an idea of the target distribution, so it tells you what tissues where the target is expressed. It can also give some indications of target epitope binding. And here, unexpected binding patterns could be due to off-target binding interaction, or it could also be due to an off-target tissue or organ reactivity. And in these cases, the unexpected staining patterns really need to be investigated further to try to differentiate if the unexpected staining is due to an off-target or just a tissue where you weren't expecting to see staining. So when looking at the two assays more head-to-head, the first obvious difference is that the MPA is a protein array. It's a cell-based protein array that uses flow cytometry, versus the TCR study is an immunohistochemistry-based assay to evaluate staining in tissues. And this gives rise to the main differentiator. In the MPA, we will precisely tell you which proteins your molecule is binding to, versus in a TCR study, you only find out which tissues that the molecule binds, and it's unclear which actual proteins gave rise to that staining. The analysis is also very different. We use quantitative analysis, and we can do statistical analysis as well, versus in the TCR, this is more of a qualitative assessment. MPA has high sensitivity. As Chris already talked about in the assay setup, we ensure screening conditions that have high sensitivity with high signal and low background. Versus in TCR, low sensitivity is often an issue. There can be high background and false positives due to native Fc receptors and IgG. The low sensitivity issue in TCR studies is really important, or has become even more of an issue for newer modalities. So the MPA is compatible to screen such things as VH molecules, nanobodies, scFv Fc fusion proteins. And there are numerous instances where customers tell us, and there's also case studies showing that these types of modalities sometimes are not compatible with the TCR studies, and where you really need to have high sensitivity to generate a robust assay. Again, in the MPA, we use native conformation of targets. We never apply fixative in any of the steps of the process, versus in TCR, fixatives and freezing of tissues often used to preserve tissue integrity, but this can lead to altered conformational epitopes on the protein level. And the timeline is different as well. MPA is high throughput. We can complete studies in 4 weeks, versus the TCR is significantly longer to complete. If you're interested more in the technical details of these two assays, comparing cell-based protein arrays to TCR, then I encourage you to read our chapter on testing for off-target binding. We were asked to contribute a chapter to the Translational Medicine book, Optimizing Preclinical Safety Evaluation of Biopharmaceuticals. This guidance book was published by 2 expert toxicologists, Joy Cavanero and Mary Ellen Consenza, and our chapter is available online. One last comparison or contrast I would like to make is, what are the two assays really testing for or testing against? So the MPA library was actually designed based on FDA guidance documents that discuss tissue cross-reactivity studies. So the tissues that are listed for this assay were selected, and then we used an RNA-seq approach to identify what proteins are in there. We use online databases and topology servers to predict what membrane proteins are actually in there. In this way, the MPA library and the proteins that are being evaluated should directly represent the tissues that are suggested to be used in a TCR assay. But here, the differentiator is that in TCR, they used normal and healthy tissues, and here you have a lot of variability between donor to donor. There's also variability in endogenous expression levels of proteins, and proteins that are expressed in a more disease-dependent manner. Versus in the MPA, we use a protein overexpression to ensure a more complete representation of all the proteins that can be found in these particular tissues. So last, let's look at some case studies that directly compare the two assays, the MPA versus a TCR. These case studies are coming from a larger study where we are testing clinical antibodies on the MPA. And so questions that we had going into this study and when we were performing this study is that, as Chris mentioned, off-target binding is commonly found in preclinical and early development maps, but what about antibodies that are actually in people right now? Do they also have off-target binding? Another question is MPA able to predict off-target binding similar or better than a TCR study? And can MPA data actually predict drug safety? So this is a question that we are trying to address because it's needed for our clinical validation for our iStrand proposal. Are there any instances where we find an off-target using the MPA where we can then link that off-target to a safety issue or adverse effects in people? Here are our approach on the experiments that we performed. We produced up to or close to 100 biosimilars of antibodies that are in all phases of clinical development and also approved antibodies. And then we screened them on the MPA to see if they had any off-targets. And this larger study is now prepared for publication. We just submitted it last week and it's under review. So if you're interested in this data and some of these discussions, then I encourage you to be on the lookout for a new paper. But back to our comparison to TCR. So what we did was we identified a few approved antibodies that actually had off-targets on the MPA. And then we compared our results to the TCR results that are obtained from BLA applications that can be found online. So this is the first case study. Here, the antibody targets a plasma membrane protein that is primarily expressed on lymphocytes. The MPA identified the target, but actually also identified two off-targets. The BLA application stated that the TCR staining was consistent with the known expression profile of the target. So this is a case where the TCR study simply missed two off-targets. In the second case study, the antibody targets a plasma membrane protein that is expressed on myeloid cells. The MPA correctly identified the target, but also identified an off-target. Here, the off-target is actually a membrane protein expressed on the same cell type as the target. The BLA application, to no surprise, stated that staining was consistent with the known expression profile of the target. So this is a great example where the TCR study really has a limited ability to identify some off-target proteins. The target and the off-target were expressed on the same cell type, and here it's basically impossible for the TCR to identify these proteins, to identify the off-target, I should say. In the third case study, this is a target that is expressed on plasma membrane and intracellular vesicles. This is an oncology target. It's very active in cancers and also upregulated in several cancers. But in normal tissue, the target has a low expression, but in several tissues, in healthy tissue, I should say. And the MPA identified the target, but also identified an off-target. The BLA application stated that staining was consistent with the known expression profile of the target. And this is then another example where the TCR really has a limited ability to identify some off-target proteins. The fact that the target is expressed both on the plasma membrane and in intracellular vesicles and throughout numerous healthy tissues made it so that the TCR really was unable to pick up any staining from an off-target. And again, if you're interested in reading more about this, then you should be on the lookout for our publication, hopefully coming out soon. So to wrap up my portion, today we talked about regulatory agencies now starting to support these emerging cell-based protein arrays like the MPA. Protein arrays are now included in CAR-T guidance documents, and our own MPA has been invited for review into the iStrand drug development qualification program. We have numerous successful IND applications that have used MPA data and not TCR data. We also discuss limitations of TCR studies and the advantages of the MPA. There are numerous technical limitations of the TCR study, and relying on TCR studies to assess specificity is really no longer supported by the majority of toxicologists. And then we looked at some case studies where the MPA was able to identify off-targets that the TCR missed. And that wraps up this portion of the webinar. Thank you to everyone listening live and anyone listening to the recording. Here is the contact information, our emails for Chris and myself. And you can also check us out at integralmolecular.com. And now I will hand it back to our moderator for any questions. Thank you.
All right, well, it is time for Q&A now. You can still submit questions using the Q&A tab on the left side of your screen. I can see we've got a lot of great questions already. We'll try to get to as many of them as possible. All right, to begin, this is for Chris. How do you ensure proteins are expressed?
Great question. Thank you. So yeah, one of a really important thing that we do is we put a V5 tag on the vast majority of our library. That allows us to take regular screenings of that V5 tag as an indirect measure of the expression of each one of these proteins.
All right, thank you. For the next question, this is for Diana. If you choose not to do TCR, then how do you know which tissues your antibody binds to?
Right, that's a great question, and we get that quite a lot. So I think the first thing, the easiest thing to do, and we do a lot, is to use a lot of available databases that are available online. So there are both RNA and protein expression databases. We use Human Protein Atlas. There's also Expression Atlas. There's a website called Big E that looks at different animals as well. So if a particular protein has a fairly well characterized, or is fairly well characterized, these websites can be very useful. The Human Protein Atlas has both gene expression and protein expression data available. It's all consolidated in the website. So it gives you a good idea of, or information on where the target is expressed. And then for less well-characterized proteins, say it's an off-target comes up on the MPA and it's not really that well characterized, then what you can also do is what's called tissue microarrays. So these are slides that have small sections of tissues on them, and you can stain them to see, which tissues express that target. And the nice thing there is that you don't have to use your own test therapeutic sample. You can actually use a commercial antibody that's been validated as a detection reagent to see where this target is actually being expressed. So if you choose to not do the TCR, there's just other approaches and ways to think about understanding the tissue expression profiles of that particular target that you're interested in.
Thank you very much. Here's another one for you, Diana. Can MPA data only replace TCR data for CAR T cells?
Right. So the quick answer would be no. It can replace it for antibodies as well. Where we are right now, the guidance document that lists protein arrays is the CAR T development guidance document. But I can speak a little bit about our customer experiences as well. We've had customers for antibody development submit a TCR for maybe different reasons. Maybe they tried TCR and they're going to get a good assay to work. Or maybe they did a TCR and they had inconclusive results and then chose to do the MPA instead. So these are just some from what customers have told us. But there's also a lot of customers actually that do both because they really want to really de-risk their therapeutic and get a really complete understanding of their specificity. So there's also the option to do both. And then one last point to make is this is where we are right now. And going forward, things might change. So in our Einstein application to the FDA, we actually proposed the MPA to be a replacement to TCR as a complete alternative. And, it's under review right now. And if it gets accepted, or approved as a DDT, then things might change in just the near future. So, it's always changing on how they're looking at the guidelines, but that's where we are right now. And we're seeing kind of a shift in how people are using it and replacing the TCR.
All right. Moving on. How does MPA perform for molecules larger than normal MAB, like a biospecific, or a smaller molecule like a cargo-loaded EV?
Yeah, great question. So essentially, we are going to screen the same way, but for biospecific antibodies, we actually get those fairly often. I think a good way to help identify where binding might be coming from or off-target interactions might be coming from would be to include the monospecific arms in as controls in the validation stage. So we would call those, isotype controls, but really just a control to help identify where binding might be responsible, what area of the molecule binding might be coming from. For smaller molecules, that's a great question. We get a lot of peptides that are typically smaller, than antibodies or even VHHs. I think we find that we going with our standard titration is usually what we like to do, up front. But if there are ever issues with that, that's why our assay setup is so important. We can always modify the titration. If the stoichiometry works a little bit better to start at lower concentrations if it's a smaller molecule. So there's a lot of flexibility there, and that is really the most important part of early discussions in the project planning stage of our pipeline.
Okay, thank you.
Yep.
Is the cell surface expression in your system always at high levels, and do TCRs better reflect the physiological expression level of targets?
So it is an overexpression system. We are transiently transfecting our cells to overexpress, one target. We're creating populations of cells that are all overexpressing one particular protein. So, of course, there's a little bit of artificiality there, but with the overexpression approach, we do have very high levels of protein expression typically. As far as TCR goes and the physiological relevance there, as we've said, there is a fixation. You're assessing binding in tissues that have been fixed, and so that can really alter native epitopes, which is really the advantage of our platform, is that we are performing our binding on these cells and these proteins that have not been fixed in any way. So I would say the MPA is more physiologically representative compared to TCR approaches.
All right. Are your proteins fused to a tag like GFP?
So our proteins, like I mentioned, are fused to a V5 tag. There's a V5 tag on the vast majority of these molecules. That's what we use for identifying or quantifying relative expression. We don't have any reporter molecule that we use in the actual screening that are on the proteins. We use a secondary antibody typically for detection.
Moving along, do you see any use of doing MPA and TCRs in parallel in order to answer the questions regarding the tissue types that the AB of interest binds to?
Definitely. I think our approach is to always do the MPA, gives you the most complete understanding of specificity, but the TCR could add to that as well. You could do the two in parallel. But the limitations of TCR are mostly because it's very costly and time consuming. So, if you have the resources to do both, that's great. But a lot of companies maybe don't want to do both. And then also the compatibility. So it's sometimes hard to get a really good signal on the TCR and a clean signal. So that's where a lot of people report the issues and tell us their issues, maybe why they tried it and they got some staining, but it's hard to interpret. So yeah, I think it's maybe great to do both, but you have to remember the limitations when you're analyzing the results or looking at the results from the pathologist, that there might be things that are missed, like we saw in in our case studies where we identified some of targets that the TCR didn't really pick up. So maybe there was some tissue staining, but the particular target wasn't actually revealed, or the cell that it was on wasn't revealed. So you can, of course, do both, but I think it's good to keep in mind the limitations of the TCR, that it won't actually tell you the protein that it's binding to. It just tells you the tissues.
Okay. If protein expression is checked by V5, do you just assume correct expression or folding?
We are not checking for the proper conformation of all 6,000 proteins. So yeah, that's relatively difficult to do. However, we are, a big part of the assay setup is using the known target of your molecule, just to make sure that we are assessing or we're observing binding as expected. A lot of molecules that are given to us are conformational dependent and or can be, at least, and so that's a very important step in our kind of de-risking of the screen itself. So no, we are not confirming the actual protein folding, but there are a lot of checks that we do to make sure that we are observing binding as it should occur.
I can also add that this is a little bit separate from our specificity profiling, but we use the MPA actually for research and development projects as well. So we do functional screens on the MPA, that on the research side, and we don't really see or have any instances where we think that the proteins aren't being functional or folding correctly. We've done like T cell screens, dendritic cell screens on the MPA, and where the readout is a functional readout. So even though we haven't individually tested every single protein individually for folding and conformation, all our data indicates that they are being expressed functionally.
How do you do validation of off-target hits?
Great question. So our validation process is very similar to the assay setup, in that we are doing a relatively straightforward binding assay using a titration of the known target, positive controls, our negative control. Our negative control is just empty vector, cells expressing empty vector, and then all of the targets that came up in the screen. So we are doing a straightforward binding assay there, and what we would consider a passing validation or indication of a true binding interaction would be any protein that is titrating at a two-fold higher than the negative control at a series of concentrations. So it's a relatively low bar, I would say, but our policy is to report more potential interactions, just because we don't want to. It's so important to not miss any type of potential low binding interaction. So that's typically our cutoff, is about twofold from the negative control.
Can you please clarify your approach for IND?
Yes, so we designed the library to be compatible, or to be a representation of the currently recommended assay, the TCR. And then we have updated all our reporting and all our data tracking to be compatible with GLP, which is often used for IND GLP tox studies. So we have an IND ready report, and then all our data tracking and auditing is compatible with that as well. Chris, am I missing anything?
No, I think that was that about covers it.
Yeah.
All right. Have you evaluated post-translational modifications in your system compared with normal or diseased cells?
That's a very interesting thought. We have not specifically addressed post-translational modifications. At least we know that some molecules can, are modified, like glycosylations or cleavage events, things like that. Based on what data we have from, again, assessing expected binding for molecules that are should be binding things that do get modified, we do see binding as expected. However, we're not doing specific assays to assess different types of post-translational modifications.
Okay, moving on. I think this one is for Chris. Is the initial MPA screening also performed on unfixed cells?
No, so we are strictly unfixed, or strictly, yeah, it is performed on unfixed cells. Sorry, we are strictly doing that. We showed the one comparison. I showed the one comparison of the fixed versus non-fixed, but that was really just a proof of concept for underscoring the point that it really is important to us to assess binding in those native protein conformations. So only unfixed cell conditions for us.
All right, and a question for Diana. For the case studies where TCR missed an off-target, were there any safety concerns found from those off-targets?
Right, so we take a deeper discussion on off-target binding leading to safety in our paper, so definitely be on the lookout for that. We discussed some indications where the off-target might lead to a particular safety concern, and then we're doing more of those studies right now. Where we are right now is a little bit more speculative, but very reasonable speculation based on the target biologies of what we have found and what we have found for those particular drugs. So that's all discussed in the review. And then outside of our work, there's also other published papers that show direct safety issues or toxicity from off-target binding to those in those drug programs. So we also discussed that in the review if you're interested more about the off-targets directly causing safety issues.
Do you have any data correlating off-target identified by MPA screening in confirmation in functional assays, for example, from the client?
Yeah, that's interesting. I mean, sorry, I can just jump in and then Dinah can add to it if I leave anything out. But typically, what customers share with us is what they feel comfortable sharing or what they feel is relevant. There have been instances where in either troubleshooting conversations or project planning conversations, they have shared functional data with us. However, this is usually at the beginning of projects, not really at the end regarding off-target interaction. But yeah, again, that's not with every project and it's not with every customer. Yeah, Diana?
Yeah, I can add a few. We have quite a few case studies on our website that talk about different interesting findings that we have worked with clients on. I know there's one case study, for example, like Chris had in his slides, where they were doing lead selection, and the two antibodies, one was clean, one had an off target, and the one that had an off target actually had they had seen biodistribution issues in mice, and the off target binding actually explained the biodistribution assays where they had found accumulation of the antibody where it wasn't supposed to be. And then a lot of our CAR T developers, what they will do is we screen an scFv for them and then tell them hey, we found this off target, maybe it was low, medium, or whatnot. But what they will do then is take that information and do CAR T testing in their own labs. So if we say binds this off target on the scFv portion, the full CAR T, they might test for binding. And we have seen, we have a few papers published on this too, where the CAR Ts maybe bind, but maybe not become fully activated by that particular off target. So that we have a list of papers on our website that kind of point to some of these examples where if they found an off target and then what to do next, assays to do after finding an off target, and maybe how the off target explained something that wasn't going right in their pipeline or in their studies. So a lot of that is on our website.
Okay, here's another question for Chris. What does the MPA IND report entail? Is it sent with the FDA package as it is?
Yeah, great question. So the IND report compared to the standard is going to be almost 3, I would say, times the size, length. That's going to include a much more detailed method section, things that really are required by regulatory bodies. So we're going to have a lot more reported data tracing, a lot. I would say a more detailed explanation of the results, kind of an executive summary of the application of the test article as it is planning to be used in clinical trials, things like that. That our standard report is typically just a representation of what the data and results that we found, but the IND is one that is tailor made to be submitted directly to these regulatory institutions.
Another one for you, Chris. Did you experience binding to off-target hits in your screen that did not translate into binding to off-target endogenous expressing cells? And if so, do you know why?
Yeah, so that we are not testing, endogenous expression or our molecules against cells that are just endogenously expressed. So I would say when we have identified off targets in the screen and they've passed our validation, again, we are using a low bar for validation, and we are using a overexpression approach. So you're going to have a large degree of protein available for binding to occur. So there is a very good chance that when we identify an off target in our screens, that if you go back and do some follow-up assays, we always encourage our customers to do follow-up assays with any results to confirm or further validate results that came out of our testing. If you're doing it in cells that are just endogenously expressing the particular protein or target that was identified as an off target, there is a good chance that it might not be biologically risky or dangerous, that type of binding interaction. But again, we are here to provide just a high level overview of what risks might be there for off target binding that our drug developer customers are not aware of at the time.
All right, moving back to Diana. Are the proteins in the MPA all based on adult tissues, or are there proteins that are expressed in different stages of development?
Right, so the library was designed based on adult tissues and adult proteins. But we actually had this question ourselves, so we looked further into it, and we did a bioinformatics analysis of it. And it does a good job representing placental and fetal proteins as well. Not as high, so the adult percentage is 94% of the adult membrane proteome, and then the fetal and placental proteins, I think, are around 90, I think between 87 and 90%. So it does a good job representing all the developmental stages. But again, it was designed based on the guidance documents that says to test for or against adult tissues.
Do you express peptide MHC libraries too?
That's an interesting question. Not at the moment. I mean, those are talks that we've had kind of internally, but I would say not right now.
Does your library include secreted proteins?
Another great question. It will very soon. So some of the proteins just at some point in their life cycle are secreted. So in our normal library, but we are in the very final stages of developing a secreted membrane library to screen against itself. So that should be. I think we are getting ready to launch that probably Q3. So that's very exciting. And if that is something that is attractive to you, for your drug development purposes, please reach out, because we would love to get some early testing with customer samples done.
What stage of antibody discovery or development should you test for specificity?
I would say probably as early as possible. I mean, we really, as we've kind of emphasized, this is a de-risking experiment or study. So before you are pouring in lots and lots of money into a potential candidate, that we've seen in IND studies that we have customers that have a molecule that's all ready to go, and they're just wrapping up some last minute or not last minute, but some late stage specificity testing, and we find a pretty significant off target that could be worrisome, and that of course can really derail an entire project campaign. So I would say as early as possible. Once you feel confident about your molecules. We have a lot of customers that come to us very, very early in their pipeline and will give us up to 20 or more molecules just for us to tell them which ones are clean and which ones have off target, so that they can just very quickly, very early on, go forward with the ones that are clean.
All right, for Diana, if MPA becomes an FDA-approved drug development tool, will all antibody drugs have to be screened by MPA for specificity, or can other methods still be used?
Well, I don't know if right now there's no guidance saying that they have to be, but if it becomes qualified by FDA, I don't know why you wouldn't. There would be many advantages to doing an MPA screen if FDA says that this is a tool that you should use for specificity testing. They will already know it. They will have evaluated it. They'll know that it gives you reliable results, and the regulatory review will be much simpler, both for the customer and for them. They'll know exactly what they're looking for. They won't have to reevaluate the assay. But unless they give a specific statement or update to the guidance documents, then you wouldn't have to, but I would recommend you do so.
And for Chris, what is the turnaround time for a standard project and an MPA plus IND project?
Yeah, so the turnaround, I would say, from the point that the molecules arrive in our hands is about 6 to 8 weeks for a standard project. Typically, we will give updates along the way. So, we like to share data as early as possible. So when we do our assay setup, our initial kind of optimization assay, we like to give data as soon as possible, just so that if there is anything that would potentially require some troubleshooting that might push the project out a little bit longer, we can do that as early as possible. So yeah, 6 to 8 weeks for the standard project, and like I said, about half that amount of time we try to aim for a tight 4 weeks for IND projects, just because we know a lot of customers are coming to us with some pretty important deadlines. So especially if there are follow-up assays that need to come out of that, we try to get that study done as soon as possible. So that also adds into the value of what you're getting too, I think.
All right, well, we are coming to the end of our time, so we only have time for one more question. I'm sorry if we weren't able to get to all the questions that were submitted. We will do our best to get back to everyone after the webinar. For the last question, have applications been filed only to FDA using MPA data for specificity, or also other regulatory agencies like EMA and MPA, et cetera? Were cross-tissue reactivity studies also included?
We have submitted. We know exactly. Yeah, we surveyed our customers, and I believe we have submitted to all of those you mentioned, and also I believe for all of those you mentioned without TCR studies as well. Actually, it's hard to keep track of everybody and all our customers, but I know we have many successes around the globe, and the different regulatory bodies have different standards for including TCR or not, and so I can't give exact numbers, but I think the general consensus is that a lot of our customers have had success with just MPA, and especially for CAR-T, as we talked about.
All right, well, thank you both so much. I'd like to thank all of you for attending this Fierce Healthcare webinar, for our speakers for participating. This webinar has been recorded. You can access the recording within 24 hours by using the same audience link that was sent to you earlier. Thank you again for joining this Fierce Biotech webinar today, and we look forward to seeing you at future events.